All right, we'll go ahead and get started. So first of all, thank you everybody for joining. Hope everybody's having a good Friday. As you just saw, we're going to go ahead and record this presentation. So today we actually have a guest speaker. We've got Dr. Jay Ronquillo. He is an NIH Data and Technology Advancement National Service Scholar for the National Cancer Institute. So a little bit about him. He's received his Bachelor of Science and Masters of Engineering in Electrical / Computer Engineering from Cornell University. He went on to receive a Doctor of Medicine from Virginia Commonwealth University School of Medicine. He also earned a Master's of Public Health and Quantitative Methods from Harvard, and then also a Master's in Medical Sciences and Biomedical Informatics from Harvard School too as well. He also completed a fellowship in Biomedical Informatics at Massachusetts General Hospital. So today he's going to give us a quick presentation, and the title of that's going to be Accelerating Cancer Research Through NCI Cancer Research Data Commons and NIH All of Us Research Program. So without further ado, I'll hand it over to him.
Thanks very much, Gage. Let me try sharing my screen. Can you see my presentation?
Yes, we can.
If I put it on slides, do you still see, is it in presentation mode? Does it look all right to you?
Yep, looks great.
Okay, great. So thanks again very much for having me and inviting me today. As Gage said, my name is Jay Ronquillo. I'm a data scholar at the National Cancer Institute at NIH. And the title of my talk is Accelerating Cancer Research Through the NCI Cancer Research Data Commons, or the CRDC, and the NIH All of Us Research Program. I'll give a little bit of background, some context and motivations for doing the study we did, talk a little bit about the published research we did related to the NCI CRDC and the All of Us program, and then some lessons learned. So first, a little bit in terms of, at a high level, in terms of the field that I'm involved in, like biomedical informatics. It's really a multidisciplinary field where you're really working at the intersection of a few different fields. And so, for example, if I were to see technology as one sphere of influence and everything related to technology, engineering, computer science, data science, digital health, and everything there. And if you see another sphere as the field of medicine, whether that's in different medical specialties, nursing, public health, population health. Biomedical informatics really is working at the intersection of those two fields. And I really see it as the practical application of different forms of technology in all those ways in a way that has that big positive impact in healthcare for patients. In terms of my background training, Gage kind of gave a good overview there, but just to kind of fit into the context of this multidisciplinary aspect of it, and from the technology side, bachelor's and master's degrees in electrical and computer engineering from Cornell University. Worked in the first part of my career as a software developer on Wall Street. I was at Goldman Sachs as a program analyst before deciding to make the plunge and go switch careers and go into medicine. Got my MD at the Virginia Commonwealth University School of Medicine. It used to be called the Medical College of Virginia. If you've seen there's a movie in the 90s called Patch Adams starring Robin Williams. And if you've seen that movie, that was kind of based on an alumnus of that medical school, which is where I graduated from. After that, I've got my MD, went up to Harvard, got my MPH, focused on quantitative methods, and then stayed on at Harvard Medical School when they had created their inaugural program in biomedical informatics. So got a second master's degree from Harvard Medical School, and then did a fellowship at Massachusetts General Hospital in the lab of computer science, where they do a lot of work in terms of clinical informatics and actually help develop some of the kind of the first databases used in some of the EHRs that you see today. In addition, in terms of my work experiences, I've pretty much been in the informatics field ever since and been able to work in different sectors, kind of cross sectors. And so some of the titles, if you are a practicing informaticist or biomedical informaticist or health informaticist, you can have worked in industry for a cancer data startup, precision medicine startup as a senior clinical informaticist. In academia, I was an assistant professor of biomedical informatics at a new medical school in Michigan, where I taught medical students as well as helped start up a graduate program in informatics. Then you can also work with the federal government. I was a medical officer a few years ago at an agency called ONC, which did a lot of work in terms of EHR adoption nationwide. And then most recently, I'm here at the National Cancer Institute, where I'm currently a data scholar. So and I kind of put it in the framework. In terms of today's presentation, in a nutshell, it's really working at the intersection of two things. And so it kind of tries to answer the question of what happens if you take the NIH All of Us research program and the NCI Cancer Research Data Commons and put them together, and what happens there in terms of biomedical research. just a little bit of background about kind of the two major initiatives that were part of the paper and the work that we published. The first is the NCI Cancer Research Data Commons, or the CRDC. In general, at a high level, it's a cancer research data ecosystem to enable cancer research in particular. And there are really two major components of the CRDC. One is data, and the second is tools and technologies. And so in terms of data, the CRDC has a lot of different kind of databases with different data modalities. We have, for example, genomic, proteomic data, imaging data, and they're all housed in different kind of databases and different portals and access. So for example, our genomic data commons has 85,000 cases, again, focused specifically on cancer, 67 different cancer sites, different type of cancer, as well as addresses 21,000 different genes, again, with a particular focus on cancer. I think the GDC, Genomic Data Commons data set, is about, I think a bit big, I think about like 2 or 3 petabytes of data in the GDC. In the proteomic data commons, 15,000 different proteins, 1 million plus peptides, houses about 29 terabytes of data. And then something like the imaging data commons has 57,000 cases, over 394,000 images related to CTs, MRIs, pathology images, things like that, and over close to 34 terabytes of data. The CRDC also has different other types of data, kind of data commons that are being developed currently. So we have something or recently released. So the ICDC, for example, is the Integrated Canine Data Commons, which is everything you want to know about cancer related to canines and dogs, so we're crossing species. CTDC is the Clinical Trials Data Commons, so everything you want to know about cancer-related clinical trial information. And then something like the CDS, the Clinical Data Service, which actually houses other data modalities that don't necessarily fall as cleanly into like a genomic data commons or proteomic data commons or imaging. So kind of a wide variety of data that you can use in the CRDC. In terms of the cloud resources, the tools and technologies in CRDC, we have actually 3 platforms that were developed over a period of time. Those are the 7 Bridges Cancer Genomics Cloud, which runs on top of the Amazon Web Services platform. One by the Institute for Systems Biology, Cancer Gateway in the Cloud, which is again for cancer-specific things that which have been built on top of the Google Cloud platform. And then the Broad Terrifier Cloud, which is also built on top of Google Cloud, and I think has a lot of similarities and overlap with the All of Us Researcher Workbench. In general, the cloud resource, the purpose is really to provide a workspace for really focused, controlled cancer data for a variety of users, people with varying levels of technical expertise. So if you like kind of graphical interfaces versus the command line type of interactions, there's something for everybody there. The purpose, again, is to build workflows, pipelines, and tools, particularly focused on cancer and using cancer data. And then for this particular study, we're also interested in using the cloud resources as a way to centralize different cancer definitions, mappings, vocabularies to help enable cancer research for other large databases as well. Don't need to say much about the NIH All of Us Research Program for this audience, but again, it was the program formerly known as the Precision Medicine Initiative back in 2015. Goal is to recruit a million or more people of very diverse backgrounds, and really to collect diverse data types, clinical data from electronic health records primarily, genetic and genomic data from whole genome sequencing, and then social, environmental, and behavioral data, like the data you see collected from the Fitbit that's available as well. And really the goal that got me excited was to kind of pull together really diverse data modalities as a way to kind of accelerate the diagnosis, treatment, prevention of a very broad variety and selection of diseases, not just necessarily one in particular. And then today, in terms of the ALS program today, we're all familiar with kind of the different tiers of data in terms of the publicly accessible data, the registered tier, the controlled access tier, with genetic and genomic data recently made available. In total, I think right now, when I looked up the website recently, over 482,000 participants, 291,000 EHRs, and then 353,000 biosamples, which now has genetic and genomic data available for researchers. And then, of course, the Researcher Workbench, which is, again, is a cloud-based platform running, I think, over the Google Cloud platform, over 1,300 active projects, of which I was fortunate to be able to be one of them as well. And then workspaces, notebooks, and the ability to build cohorts within the Workbench. And so those are the two major kind of initiatives and projects. And so in the work that we were doing, we actually saw some opportunities to be able to bring those two things together, the CRDC, the Cancer Research Data Commons, with the NIH All of Us Research Program. And so the way we kind of saw it is that the All of Us Research Program is solutions looking for a problem, whereas the Cancer Research Data Commons are problems looking for a solution. And so definitely potentials for synergy in the sense that the All of Us Research Program is a disease agnostic database which has information about many different types of conditions over a course of a person's or patient's life, and the researcher workbench to be able to access the data. On the other side of the spectrum, you have the CRDC, which is a very cancer-specific oncology-focused cloud resource, set of cloud resources where you have cancer data, cancer tools, and definitions. And so the way we kind of saw it was the CRDC is a way of being able to unlock the All of Us Research Program's data in a way for particular scalable cancer research. And that's what we wanted to do for this work. And so we published a paper earlier this year, actually in January, published in the Journal of Clinical Oncology, Clinical Cancer Informatics, talking about the landscape of genomic testing for patients with cancer using the All of Us database as the primary source of data and the Cancer Research Data Commons as the cloud resource to generate the definitions and mappings to focus on cancer. So the purpose of the goal, so the question we really wanted to answer for the study that we published was, for patients with cancer, what is the current practical landscape of precision medicine and genomic testing very broadly across the country based on the data that's currently available? And so in terms of our methods, what we looked at really focused on identifying patients with cancer, very broad definition in terms of lots of different types of cancer. Wanted to assess genetic and genomic testing that's currently available in the All of Us program, as well as assessing the type and the nature of the clinical genomic results that came back from this database. In terms of the way we defined cancer, we actually looked at so as the All of Us database, as you know, in terms of describing patients, their condition and diagnosis, they're in the form of ICD codes, ICD-9 and ICD-10 codes. And so we wanted to get from ICD-9 and ICD-10 codes to get to the actual cancer types associated with that in a very structured, formal, defined way. And so we actually started by using the NCI's SEER program, Surveillance Epidemiology and End Results Program, where it has definitions for various cancers and cancer sites. They're currently mapped to ICD-03 codes, which is a special type of ICD vocabulary focused on cancer specifically. And so we just converted those mappings from ICD-03 to ICD-10 and ICD-9. We also use something called the GEMS mappings, which are general equivalence mappings between ICD-9 and ICD-10, supported by the Centers for Medicare and Medicaid. And so once we defined cancer in that way, we defined the cancer cohort as anybody who was included who had greater than or equal to 2 documented cancer diagnosis instances, which in this case means at least 1 ICD code and then 1 reported cancer from their personal medical history from the surveys that patients who enrolled in the All of Us program report. The reason we did this was we felt that in order to get a kind of a fairly stronger definition and be sure that you're not getting false positives or underreporting or kind of misreporting of data, we kind of did this way to kind of define cancer. So the idea being that if you're finding an encounter of a diagnosis in the EHR, it's more than likely that the patient will also report that as having reporting that cancer as well in their personal medical history survey. You can also include people with 2 ICD codes as well, but again, trying to get a more robust definition of cancer for this particular study. In terms of defining genomic testing, we focus on LOINC, the LOINC vocabulary. LOINC stands for Logical Observation Identifiers, Names, and Codes. It's basically a vocabulary focused on defining in a structured way actually laboratory tests in general. And so for the LOINC vocabulary, we pulled out all the codes focused specifically on genetic and genomic laboratory tests and focused on particular categories. And so this included things like deletions and duplications, translocations, nucleotide repeats, as well as things like cytogenetic reports, molecular pathology, next generation sequencing, even whole genome sequencing, to try to capture what is the current state of genetic and genomic testing, at least from the kind of codified perspective. And then from those results, we also investigated the clinical relevance of those coded results in terms of how easily could you extract findings that are either clearly positive or clearly negative from those results. In order to pull this all together, we built an informatics pipeline focused on cancer for the study. And so first step was to actually load all these external files. So the LOINC vocabularies, ICD-9, ICD-10 codes, the SEER database, to be able to get an idea of those definitions. And we then created within the Cancer Research Data Commons using a Jupyter Notebook running R, I think version 4.0.3 at the time. And we created cancer mappings and definitions to define cancer, to define genetic and genomic testing and extract those. We then use those definitions within the All of Us Research Workbench. So again, using a Jupyter Notebook running Python, we extracted the cohort data, specific patients with different types of cancer, genetic and genomic tests. And then using a third notebook, the second one in the All of Us Research Workbench, we actually did the analysis where we integrated, analyzed, and visualized the data, again, pulling some additional definitions from we created in the CRDC, and also the data that we pulled from the All of Us Research Workbench from the controlled tier data. or registered to there. In terms of what we actually found, we're able to find, this is the first table summarizing the characteristics of participants by cancer status. Overall, we've in the database at the time, we found about 5,600 in the cancer cohort participants, and then in the non-cancer cohort, about 309,000 patients, you can see. And you can see here, there's kind of a diverse group of people in terms of age. Generally, they tended to be older. In terms of sex, at least sex reported at birth, primarily 60% female, both in the cancer and non-cancer cohorts. And then some different kind of differences in terms of race and ethnicity as well. You can see that in the non-cancer cohort, it was about 50% white and then about 25% Black, African-American, and Asian. Whereas in the cancer cohort, it actually was a little bit different in terms of instead of 50% white, you had 86% white, so a little bit of a higher proportion there for doing that. And so it just kind of highlights the differences once you start to filter down the All of Us program by different conditions and diseases like cancer. In terms of the general cancer categories, you can see here we have kind of a wide variety of different ones based on the SEER categories. Again, the key takeaway here is you can see some of them are things you would expect based on kind of what's reported in the CDC. So for example, breast cancer is very common, prostate cancer. I think breast has about 2,000 plus patients, prostate cancer close to 1,000. And then you have also other ones like melanoma, leukemia, and lymphoma, head and neck, colorectal, and things like that as well. So kind of a broad spectrum of cancer, hundreds and potentially thousands of patients for each given cancer category as well. When you're looking at genetic and genomic testing, kind of the same thing. For the cancer cohort, there were 295 patients, making up about 5% of the cohort who received genetic and genomic testing, whereas for the non-cancer cohort, there's about 2% of that population. In total, about 34,000, close to 35,000 genetic tests were ordered. And as you can see here, the most common in terms of the testing categories, you have kind of a wide variety, but relatively few patients in most categories, with the exception of maybe the gene mutation, the broad gene mutation category. So for example, in the cancer cohort, 216 patients with gene mutations. These are a total of about 27 different types of unique genes. And these are on the left, you can see the most common ones. HBB is the hemoglobin beta subunit. F5 is for coagulation factors. F2 is another gene for coagulation factors. JAK2 is one that's common for different kind of blood cancers. And then MTHFR is used as related for drug interactions, particularly for methotrexate, so another chemotherapy drug there. And then there's also some testing for cancer-focused next-generation sequencing. In terms of the genomic test result characteristics, I think the key takeaway here is that there are about 55% with structured clinically usable findings. And so, which means that in the structured data, in terms of looking at the results, you can see them something that's clearly indicating a positive finding or a clear negative finding. So positive findings listing things like telling you it's positive or carrier was detected, abnormal result, or homozygous heterozygous, whereas a negative finding is something that's saying it's not detected, normal, low risk, or wild type. Which means the other half is what we kind of classified as other, things that aren't necessarily clinically usable or understandable. So that might mean that there is no result available at all. There might be unstructured text or a finding that doesn't seem to make sense by just kind of random numbers, for example. And so that's kind of the characteristics in terms of genetic and genomic test results. So what does this all mean in terms of what are the findings? So some of the key takeaways in terms of the implications for precision medicine. I think the first one is saying that genomic testing is still relatively rare if you're looking at particularly your database, although you can see that the cancer population, cancer cohort, is tested a little bit more frequently than the non-cancer cohort in terms of 5% versus 2%. These numbers by themselves also could potentially be underestimated just because of when you're talking about EHR data, there's kind of this idea of missingness that needs to be factored into. Also, as issues in terms of interoperability. Sometimes, for example, the data feeds into one EHR, but if a patient receives care in another EHR, that may not be transferred to the EHR in the All of Us program. And then things like lab results being returned. So sometimes when the genetic tests are being sent out to labs, some of those results may not, again, be received back by the EHR electronic health record, or they may not be received in a way that's in a structured way that can be extracted. And I think the key takeaway from this is that cancer is a potentially valuable use case for the All of Us program, and that you can, if you look at the previous few slides ago, for any researcher looking at particular cancer types, you can actually find hundreds, potentially thousands of patients to do your cancer research in the All of Us program. And I think that's an important finding. The other step is that most tested genes are pharmacogenomic biomarkers for FDA drugs. And so if you're actually to look on the FDA website, they have a table of pharmacogenomic biomarkers that have been listed or indicated in the FDA drug labels. I'm using the labeling in terms of recommendations. And I think that's an important finding as well in terms of when you're looking at genetic and genomic testing, there are multiple potential applications for using genetic testing. That includes things like carrier testing, diagnosis, prognosis, as well as kind of things like pre-implantation testing, prenatal testing, and newborn testing. But by and large, if you were to look at the data here, and the EHR data in the Oliver's program as a practical representation of what data is kind of being practically used right now, pharmacogenomics seems to be potentially the low-hanging fruit when you're talking about adopting it for precision medicine. And then also, just about 50% of the genomic results are structured and clinically usable. And I think this is important to highlight the importance of further taking the data, standardizing, harmonizing, integrating, and aggregating that data in order to be able to scale that up. And I think when you do that further standardization, harmonization, it really opens up opportunities for applying things like artificial intelligence and machine learning and deep learning, as well as building clinical decision support tools within the AHR or outside of the AHR to help scale up, again, kind of precision medicine, personalized medicine, individualized medicine. There's also, I guess, to highlight, there's the value of using the cancer research data commons and informatics tools for cancer research. Again, the CRDC was able to, using these definitions, be able to unlock large studies for cancer-related research. Again, you're in just a short amount of time, you're able to make hundreds of thousands of patients' data available, as well as to study over 15 different cancers, 34,000 different genetic tests, just by defining and mapping and looking at data in the correct way. So unlocking the data using the CRDC is, for all of us, is potentially valuable. I think also being able to accelerate the publication of cancer research. I think using the CRDC with informatics and the All of Us program combined really helps to streamline that process a fair amount. In terms of, for this particular paper, it took about 2 months to actually perform the analysis, meaning to define the study, build the pipeline, and actually write out the paper. So 2 months for doing the analysis. And then the peer review process also was fairly quick in terms of the time you actually submit the paper to the time it's actually accepted for publication. Only took about 2 months for us as well, simply because most of the feedback that the peer reviews give really involves updating, revising, or expanding the algorithms and the pipeline that you build for studies like the All of Us program. And then additional 2 months just for publication once it's actually published. So again, within a very short amount of time, within less than 6 months, you can actually come from start to finish, get a peer-reviewed, high-quality paper published using in cloud resources from the CRDC and the All of Us data. And again, in terms of research productivity, over the past, we estimate about the last 8 to 12 months, we're able to publish 3 different papers, again, focused on CRDC and the All of Us research program, and give 4 presentations in conferences ranging from very medical conferences focused on cancer, kind of the medical side of cancer, as well as to more informatics and conferences like the Odyssey Conference and the Global Alliance for Genomic Health, as well as conferences like the focus on precision public health as well. So again, there's a broad spectrum of opportunities, again, of using the All of Us research program and the Cancer Research Data Commons together to do cancer research. In terms of future opportunities for the All of Us program and where that may fit, I think there are probably 2 ones that kind of stick out from my perspective. One is a project we're working at NCI right now called the Cancer Data Aggregator. And so in the same way that the All of Us research program integrates and aggregates and harmonizes electronic health record data, as well as Fitbit data and other data, and brings that all together for research, the Cancer Data Aggregator is doing that for cancer-specific data. So genomic data, proteomic data, imaging data, and again, integrating, aggregating, and standardizing and harmonizing that as well. And so that's one potential opportunity for potential synergies, I think, going forward. The second one is a project called the NIH Cloud Platform Interoperability Project, or the NCPI. And so the same way that All of Us is aggregating EHR data and the Cancer Data Aggregator is aggregating cancer-specific data, the NCPI project is actually aggregating the aggregators together. And so you're actually pulling in large cloud-based platforms and the data associated with a lot of initiatives going on. So the NCI's initiative is called the Cancer Research Data Commons. The National Heart, Lung, and Blood Institute has one called the Biodata Catalyst. The National Human Genome Research Institute has one called the Anvil Project. And you also have one from other organizations as well. And so being able to pull this data again at a very large scale to conduct large-scale biomedical research opens up, I think, important opportunities. And again, is another opportunity for something like the All of Us program to potentially participate in as well. So in conclusion, the All of Us program provides some very practical insights into the landscape of precision medicine. And I think understanding what the current state of precision medicine is will help us to understand kind of where we need to go and what we need to do to get to the precision medicine we want to get to in the future. The Cancer Research Data Commons is also capable of unlocking and accelerating cancer research. And I think in terms of being able to make precision medicine a practical reality, that'll involve projects like the CRDC, projects like the All of Us program, where it's focused on kind of integrating and aggregating that data, addressing interoperability from multiple perspectives, and then also building the informatics tools to actually pull the data together, perform the analysis, and then share that data with large populations of people, patients, as well as helping to scale that up for researchers as well. So I just want to acknowledge my collaborator and co-author, Dr. Bill Lester from the Massachusetts General Hospital and Harvard Medical School, the National Cancer Institute as well, Center for Biomedical Informatics and Information Technology in the Data Ecosystem branch, and then Office of Data Science Strategy, as well as all of us participants, without which this research wouldn't be possible. So, thank you very much.